Okay. Hi, everyone. I'm Hans, a postdoc at Diamond Light Source, and it's my pleasure to chair the keynote lecture of the study weekend. We have the honor of welcoming Ivan Nicolet. He's famous for discovering the first iron-only hydrogenase. Nowadays, he leads the metalloprotein group at the Institut de Biologie Structurale in Grenoble. But in his spare time, he enjoys reading European comics. But today, he's here to tell us about metalloproteins, the best kind of proteins. I might be biased. But Ivan, the floor is yours.
Thank you very much for the kind introduction. And I also want to thank the organizers for the kind invitation. That's a real honor to be here today. So I will try to introduce you to this field of metalloproteins for those who don't know, and the others, they might be more aware of that. But I will start with a short introduction, maybe this way. Okay. So what I call in my group, what we call metalloproteins. So what we call metalloproteins, if we are interested in proteins that contain transition metals, and the definition of transition metals is the molecule, the elements that belong to the subshell. Sorry, that's normal. Okay. Yeah, the subshell elements, and amongst all these metals, only a few of them are present in proteins, which are listed here. And we are interested in my group only on the properties of these metals and the relationship between these metals and the activity of the proteins that contain them. They are all essential for life, but in the meantime, they are highly toxic if they are at a higher concentration. So why transition metals? Because they can have different geometries depending on the ligand coordination. And here I present you the main ones. That makes very peculiar interaction with the proteins. And also what is interesting is that they can have different redox states, but they can also change their coordination depending on the redox state and on the surrounding, meaning that you can have bonds that can be broken or made, and the substrates in enzymes can interact with these metals and that can be activated. And in the meantime, these metals can also play a role in electron transfers. So you can also have redox-based reactions. But you can also have non-redox-based reaction, as I said. The properties of these metals are modified by their ligands, and also they provide to the proteins they are in new properties and new chemistries, which is what we are interested in. So in my group, we are more focused on iron, sulfur-containing proteins. Here are a few of the metal centers that you can have in proteins. The more common ones are the 2-iron and the 4-iron phosphate clusters, but you can have more sophisticated one, including other transition metals such as nickel here or molybdenum there. These proteins are involved in very essential reactions in life. This one is the active site of the carbon monoxide dehydrogenase involved in the CO2 fixation. That one is the one I worked on during my PhD long ago. This is the H cluster. This is the hydrogenase active site. So it's involved in the H2 production or oxidation. And that's one which I really like, because to me it's one of the most beautiful cofactors, metallocofactors in life, is what we call the FeMoCo. That's the active site of the nitrogenase, which is involved in nitrogen fixation. The resemblance of these metallocofactors with minerals has led long ago to a theory about mineral origin of life, and I will not go too much into the details, but if you connect hydrogen and carbon and CO2 fixation, you can make acetyl coenzyme A through the Wood-Lungwall pathway. And the active site of these proteins resemble this mineral, for instance. And so it has been proposed that before proteins, this chemistry could have happened on these surfaces. Nowadays, these metalloproteins are also essential for other pathways, such as photosynthesis. And this leads to the appearance of oxygen and respiration. And these two reactions are a major invention in nature that led to complex life on Earth. Photosynthesis leads to the appearance of oxygen, and oxygen is a strong oxidant that is toxic for metalloproteins. And here I will briefly show that we are lucky because oxygen can exist in two main states, the ground state that we call the triplet one, and the singlet state. This one needs to be activated, but this one is very, very active. Fortunately for us, this one is not very reactive. That's why we can exist and we don't burn in contact with oxygen. But still, oxygen can react with electrons, and it leads to reactive oxygen species. And you might know that they are harmful for life. So, as I already said, oxygen is not the strongest oxidant in the process. But if you provide one electron, you can have the superoxide radical, which is the most stronger oxidant. Oxygen should react mainly through direct contact, so inner sphere or electron transfer, while the superoxide can react with longer distance to oxidize the molecules. It can dismutate to produce another reactive oxygen species, which is hydrogen peroxide. This is also a strong oxidant, but not that toxic. What happens is that hydrogen peroxide is known for the Fenton reaction because it reacts with iron 2, leading to this one, hydroxyl radical. And this one is the nastiest guy in the group there because it's one of the strongest and highly reactive species. It reacts very readily with all the surrounding molecules, and that causes damages to lipids, proteins, and DNA. With
you to use the on-screen pointer.
Okay, because we don't see it. Okay.
The people online can see the
okay. Sorry for that. Okay. Okay. Regarding the iron sulfur clusters and their reaction with oxygen, it has been shown that oxygen can abstract one electron to a 4-sulfur cluster, leading to the production of a superoxide, and a 3+ 4-sulfur cluster, and then the superoxide can also react with the 4-sulfur cluster and the hydrogen peroxide as well. And all these lead to a 3 iron 4 sulfur cluster with the release of 1 iron 2 that can also react with superoxide or with the hydrogen peroxide to do Fenton reaction. And the problem is that this 3 iron 4 sulfur 1+ cluster readily decay into a 2 iron 2 sulfur cluster. And this is spontaneous and very fast. And so overall, if you have one oxygen molecule, you can break up to 4 iron 4 sulfur clusters. But these species also react with the ligands around the clusters. These ligands are often cysteines, and you can have oxidation of these cysteines through the formation of cysteine persulfides because there is the release of sulfides during the disruption of the cluster. So this is why in my group we are using glove boxes to prevent the damages of our proteins when we are expressing them, purifying them, crystallizing them to do the studies. So we have a bunch of glove boxes to do the expression, purification, crystallization. For instance, we have a crystallization robot. I'm a bit of old school compared to the different techniques that have been presented over this day, but still we try to do the best we can on the analysis of the function of these proteins. So we have also an automated arm to screen for crystallizations plates over time, and we recently implemented a Vitrobot to do cryo-EM as well. And we can also do some stop flow to do some kinetics under anaerobic conditions. Okay. Sorry. Okay. The proteins I want to talk about today belongs to a superfamily of proteins that are called the radical S-adenosyl L-methionine, or SAM enzymes. These proteins catalyze very difficult reactions. It's a broad, maybe the broadest superfamily of proteins, with over 850,000 sequences known. They can catalyze over 70 different reactions that are known today, and they all share the same structural features. One is they have a conserved CX3, CX2C motif that binds a 4-phosphate cluster, and they also share a structural core to accommodate this cluster and to bind the substrate. But the chemistry that is catalyzed is very different, and I will so the trigger is the reduction of the cluster to cleave the S-adenosyl methionine molecule into a 5' deoxyadenosyl radical, and this radical will react with an organic molecule, or in some cases non-organic, but I will show you later on, to trigger the radical reactions. In most cases, it's a hydrogen atom abstraction to activate the substrate and then to do the catalysis. Here, just to show you the diversity of these proteins, these are a bunch of different reactions that these can catalyze. So still the same trigger and the same structural core, but they can do these. And more recently, some of the subfamily of proteins have been discovered as being involved in the post-translational modification of peptides, giving them antibiotic properties. Today, I will speak more about the radical SAM enzymes that are involved in the metallocofactors. There are 3 proteins known: HYDE-E, HYDE-G, and NIF-B. So the 2 first are involved in the synthesis of the H cluster for the iron-iron hydrogenase, and the last one is involved in the maturation of the nitrogenase cofactor. So here, the H cluster and the FeMoco. So first, the iron-iron hydrogenase active site assembly. So iron-iron hydrogenase is a protein involved in the reversible oxidation of molecular hydrogen, leading to protons and 2 low potential electrons. This is possible because of the active site that is buried in the protein matrix, and the reaction takes place here. And the synthesis of this cofactor is not spontaneous, starting with iron and sulfur and carbon and nitrogen. It needs a dedicated machinery. And this machinery is divided into 2 parts. The housekeeping machinery is building the 4-phosphate cluster, and the HYDE machinery, the one that is specific to the hydrogenase, is dedicated to the 2-iron subcluster assembly. What has been shown over time is that the precursors for these are iron and amino acids. This was a bit surprising, but it's an interesting feature in terms of evolution. this machinery. And in organisms, this machinery that is called hide is often in operons. And this is in the sulfovibrio vulgaris Hildenburg. You can have the machinery with Hig, a radical same enzyme, aspartate ammonia lyase, which is involved in the production of the nitrogen here, and Hig, another radical same enzyme, and this Hig-F is a scaffold protein that is involved in the insertion at this later stages of the assembly. So very briefly, Hig-F, it contains a 4-phosphate cluster. It can accommodate the precursor of the 2-iron subsite, and it transfers that into the hydrogenase, leading to the active enzyme. Ig. So we started long ago on Ig without knowing anything about the activity of the protein. The only thing that we knew was the sequence and the fact that it was involved in the maturation of the hydrogenase. And one striking thing is that it resembles Thi-H, another radical same enzyme that is involved in the synthesis of the thiamine vitamin. The difference between the two is that Thi-H contains only the radical same domain, and Hig contains an extra domain at the C-terminus with cysteine-rich extra motif. So briefly, to summarize what both protein did, they used tyrosine to break this bond to make the dehydroglycine here, and while Thi-H is transferring that to the next steps to provide the N and C of the thiamine ring. Hig is splitting dehydroglycine into carbon monoxide and cyanide to provide the precursors of the iron-iron hydrogenase active site. And it has been shown by EPR, by electron paramagnetic resonance spectroscopy, a technique that detects single electron containing molecules, that paracrasyl radical is produced in both proteins. So we did a truncation of the protein, removing the C-terminal part, and we did Sachs under anaerobic conditions. And we show that when we remove the C-terminal part, Hig behave the same as Thi-H, as it produces dehydroglycine and no more cyanide and carbon monoxide. We determined the crystal structure of one of the Ig protein, and in the meantime, in Southampton, our colleagues determined another structure, and combining the two structures, it has been possible to show that first, the second cluster, so the C-terminal domain of the protein contains an auxiliary cluster that is composed of a 4-phosphate cluster and a 5th iron that we call the dangler iron. And this iron is connected to the 4-phosphate cluster through an amino acid that turned out to be a cysteine residue, not residue, a cysteine ligand that binds this iron. And there are two water molecules that are exchangeable ligand, and this iron is also bound to the protein through an histidine residue. So analyzing the structure, we were able to show that the protein contains 3 different subsites. The first one is dedicated to tyrosine splitting. The second one is dedicated to dehydroglycine splitting. And the third one is where the auxiliary cluster is, and it's dedicated to carbon monoxide production. We don't know in great details the mechanism, but we have some information. The first work that we did was to simplify the system by studying the Thi-H protein, that is the radical same domain of Ig. So it corresponds to this part. So we determined the structure of Thi-H from Thermoanaerobacter carboxyliborans in complex with its substrate, and 5'-deoxyadenosine to mimic some cleavage product. And we were able to show that the C5' atom that holds the radical is facing the nitrogen atom of tyrosine. showing that the hydrogen atom that will be abstracted is from the nitrogen atom of tyrosine. And unexpectedly, we show that the tyrosine bonds to the protein is completely deprotonated. And this is important for reaction because this nitrogen here is facing an arginine residue, and this carboxylate is facing a carboxylate as well. And this phenyl hydroxyl group is interacting with 4 different hydrogen bonds. That is four different hydrogen containing moieties, showing that it cannot be protonated. And we did some molecular dynamics showing that indeed it has to be deprotonated to be to bind the way we observe it in our structure. And in fact, it's important for the reaction because upon hydrogen abstraction, in order to be able to cleave this bond, it has to be deprotonated here to produce this para-hydroxybenzyl radical that has been detected previously by paramagnetic resonance spectroscopy, as I mentioned. So now we understand a bit more the first step of the reaction, but what about the second step of the reaction? What we know is that between the tyrosine binding site and this region here, there is a ring of hydrophobic residue that disrupts the hydrogen bond network of the water molecules that can fill the wall cavity. And there is a chloride ion that we observe in both structures, suggesting that this can bind a negatively charged molecule. We don't know the reaction for the hydroglycine splitting, but we suspect that this is where the cyanide, the produced cyanide should bind. And the same for the last part. It's very difficult to access later stages of reactions, and so far we don't know how carbon monoxide is produced. The only thing that we were able to show is that if we remove this histidine residue, we disrupt the 5th iron, and then we produce, instead of CO, we produce formate. Still, if you get the enzyme and you put 1 tyrosine as substrate, 1 SAM molecule to trigger the reaction, and 1 electron, you can make 1 turnover, and you can detect on the auxiliary cluster a new species that has been termed complex A, and through isotopic labelings, it has been shown that this molecule contains 1 carbon monoxide and 1 cyanide. And if you repeat the reaction with another equivalent of tyrosine, then you have another species that is termed complex B, and that corresponds to a 4-iron phosphor cluster in the reduced state with cyanide bonds to the iron, and the release of this part, and that has been proposed to be the precursor for the synthesis of the 2-iron subsite of the H cluster. And our colleagues in the United States were able to synthesize a mimic of this molecule, and through different labelings, they have shown that the iron, the sulfur, the carbon monoxide, and the cyanide all end up within this 2-iron subsite. So now height E. I've been working on this protein for about 15 years. When we started, we didn't know the substrate, and it was easier to get the structure of the protein than to get the function that we still don't really know. The protein, by soaking the crystals with iodide solutions, we were able to show that the large cavity that goes from the top of to the bottom of the beta barrel here contains 3 anion binding sites. We also were able to show that in this region, thiocyanate can bind. At that stage, we didn't know why, and we still don't really know. But again, as in Ig, there is a ring of hydrophobic residues that splits the cavity into two different pockets. And the active site where the radicals-based chemistry should take place is there. But if we mutate residues in this region, we can also impair the hydrogenase maturation. Okay, so in order to try to find what the substrate was, we used the fragment lead approach by soaking our crystals with different type of molecules, small molecules, larger molecules, but small molecules. And we ended up with the fact that when mixing L-cysteine with pyruvate, we can make this thiazolidine ligand, and this binds to the protein in front of the SAM molecule. So it could be a substrate, and changing this pyruvate by other molecules, we're able to show that the protein can bind different types of ligand always the same way. And what is interesting is that the C5 prime atom that holds the radical is facing the sulfur atom of this thiazolidine. So we knew that this wouldn't be the substrate of the protein because it cannot make anything for the maturation of the dehydrogenase. But yet, soaking a crystal with a thiazolidine labeled with selenium here, triggering the reaction by soaking the crystal with a reducing agent, we are able to monitor the reaction within the crystals. It's pretty slow, so we were able to freeze by hand our crystals in the glove box and take them out of the glove box to go to the beamline. And so after 2 hours, what we observed is the formation of what seleno S-adenosyl L-cysteine or selenocysteine, and the appearance here in this region of a chloride ion. We showed that in solution, this is happening as well. We form SAC or SE SAC, depending on the ligand that we use. And we monitor the reaction at different time points. And we calculated different Fourier maps showing that SAM is cleaved and the C5 prime is attacking the sulfur atom of the thiazolidine ligand, and the pyruvate part of the thiazolidine is disappearing here in the electron densities. Okay, and we wanted to know what happens to this moiety, so we prepared a labeled thiazolidine using a pyruvate where the methyl group is labeled with a C13 atom, and we monitor by NMR what happens to this molecule over time. And this is the pyruvate from the beginning. And this is the substrate, or the non-natural substrate, the thiazolidine. And what we showed is that upon reaction, we release pyruvate. So when it disappears here, it produces pyruvate back. So we were able to propose this mechanism with some cleavage from one electron reduction, and everything is aligned. So the C5 prime is formed, it attacks the sulfur, it produces a radical intermediate that is oxidized, and then this is hydrolyzed, leading to the SAC that we observed in our crystals. But why I'm talking to you about that is because if you look at the thiazolidine that we observed in the protein, it looks like the molecule that has been proposed to be the product of Ig. It's a bit similar to the thiazolidine. And in fact, our colleagues in the United States, by synthesizing a mimic of this molecule, they were able to show that if you soak your molecule into a Ig solution and you trigger the reaction, you monitor by electron paramagnetic resonance spectroscopy over time the reaction, and they detected one new species after 10 seconds and another after 10 minutes, and using different labelings and under advanced techniques, EPR techniques, they were able to show that propose these two structures for these two intermediates. So it looks like the product of Ig is the substrate of IE. So back to our lab, we soaked our, we co-crystallized our protein with okay, we'll finish with that. With SynB, we got the structure of the molecule bound to the protein, and this helped us to determine the correct configuration of the iron there. And we wanted to show that this mimic is really the mimic of the product of Ig. So we used Ig and we inactivated IE. We triggered the reaction of Ig with tyrosine, SAM, dithionate as the reducing agent, iron on cysteine to produce this molecule. And we we mixed the two molecules together and we crystallized IE, and we got the structure of the enzymatically produced complex B that corresponds exactly to what we had before. And so why it is interesting is because complex B compared to the thiazolidine, the sulfur atom here is nearby the C5' atom, like in thiazolidine. So we trigger the reaction in the crystals the same way as we did with the thiazolidine, but unfortunately, this time, what we observed is the missing part, which is the iron part. And I remind you that we were expecting this type of molecule, but we only see the ligand here. And in fact, we tried to do the same experiment as with the EPR. This time we trigger the reaction in solution before crystallization. And the reaction takes place here, but what we observed is in this region, we observed a new feature in the electron density. And this feature has been interpreted as an iron atom because we recorded the anomalous signal before and after the age, showing that this is an iron that is bound to this methionine sulfur atom. These two ligands are in hydrophobic pockets that we model them as carbon monoxide. This one is it's establishing hydrogen bonds, so it's a cyanide. And after refinement, this is the electron density of this penta-coordinated iron species. That was completely unexpected. And the question is whether this molecule can be the substrate. That's just to remind you that normally we should have a sulfur here. So the question is whether this water molecule here could have been a sulfur that has been lost over the time course of the crystallization. And I told you that the protein binds cyanide, thiocyanate here. And when we superimpose our structure with our previous structure with thiocyanate, we have the nitrogen atom overlapping with the nitrogen atom of the cyanide ligand and the sulfur atom ending in the region where we have this X ligand. So it is possible that this is a decay of the product of the protein. And I will, we observed in the cavity that this complex B binds on top, facing the C5, the 5' GA, but we have a methionine residue that is conserved, that is in between the two pockets. And in our structures, we see that this protein, this methionine has two conformations, and it can be a relay to transfer the iron species from one pocket to the other. And I will switch that here, and I will just, acknowledge the people who did the work. Yeah. Okay. So I want to thank all the people from my group who did all this work, and the collaborators and the funding agencies. And I thank you for your attention.
Thank you very much. That was awesome. Do we have any questions from the room? Over there.
Hi, it's a really nice talk, and I'm quite interested in that because I've been working on ion supercluster proteins, including B family DNA polymerase and some helicases. So I have a technical question to ask. So I was wondering if prevent oxidation during DNA production is really matters, because clearly not all the lab has the fancy facilities as your lab does to prevent, the octades, to reduce the octade. Yeah.
Yeah, I understand.
Yeah.
It depends on whether the cluster is stable over time when you purify it aerobically.
So I was wondering if there is any difference between different type of ion supercluster.
Yes.
Oh, that's great. Yeah.
First, the 2 ion supercluster are more stable than 4 ion superclusters. And depending on the environment of your 4 ion supercluster, because in your case, it's 4 ion superclusters.
Yep.
Depending on the environment, they might be more stable in some proteins than in others. But clearly, with your proteins that interact with DNA, there is a decay over time. So it depends on what you want to measure. I know that some people who are working in similar projects, they show that if you don't have the cluster, you can have an artifact with one single turnover or something not very efficient. And if you have the cluster, it works better. But so on some other proteins, it doesn't matter. So because it can be replaced by zinc.
So which means if I'm going to do some biochemistry-based assays, it might not be that matters because the protein is still active. However, if I'm going to do some structural-based analysis, it could be an issue, right?
Yes, it can be. It has been shown for the SARS-CoV-2 polymerase, because the first structures that have been solved show 2 zinc fingers, and it turned out that it's not zinc fingers; it's 2 4 ion clusters that are important for the reaction and for the activity. And the people in the United States will show that. They show that if you target with a radical species these clusters to disrupt the infection. So it matters. What I suggest you is that you measure your activity in aerobic and anaerobic conditions, and if it works the same, then it doesn't matter. But if you have a difference, then you have to switch to anaerobic environment. You're welcome.
Are there any questions on Slack? Any in the audience? Back here. Thank you, Ronan.
I don't know if it would, I mean, you.
Oh, sorry, can you introduce yourself and your affiliation so that people online know?
Vladek Minor, University of Virginia. I do not know, you put tremendous effort into this research, and I assume that all your metals are correct. Although in PDB there are many metals which are incorrect. What if somebody would, and this is the question partly related to the next session or next speakers, what you would do if somebody would show you that you have incorrect metal? What would be your action? Not reaction, but action.
If I had an incorrect metal bound to my protein, that happens sometimes. It really depends on whether this is important for activity or not, because sometimes you can have metals at the surface that binds to the surface, but they are not important for activity. In my case, I know that the protein works with iron sulfur clusters, so it must be 4 iron and 4 sulfur. And I know that now, from what we have shown, that the substrate is an iron-containing molecule. And from the anomalous signal, we are sure that it is an iron that we are tracking in the reaction. In these proteins, I have not seen any other metals, but that can happen. And again, if it's important for the activity, then I would work very hard having the right one.
But would you correct your previous deposit to PDB?
Oh, for sure. Yeah.
Okay, thank you.
Thank you very much. Shall we thank Ivan once again for his great talk?